FLLIT is an algorithm that calculates the body and leg claw positions of a tracked animal. And this is useful for characterizing the circuitry that underlies both normal and diseased walking behaviors. The unique thing about FLLIT is that it does not require any user-annotated datasets for training and is hence fully automated. FLLIT also automatically analyzes the track data, producing a series of gait measurements, gait plots for visualization, and a track video. Demonstrating the procedure will be Animesh Banerjee, a postdoctoral colleague from my laboratory.
Before beginning an experiment, confirm that the recording station has a high-speed camera with a stage over it to hold the arena chamber. Place infrared LED lights at the top of the stage with a diffuser between the camera and the sample. About 40 minutes before beginning the recording, transfer the flies into an empty vial on ice for 5 to 7 minutes. While the flies are being sedated, use water and a wipe to clear the arena and glass slides of the chamber and tape one of the glass slides below the chamber. Using a clean brush, transfer one anesthetized fly into the chamber and secure the chamber with a second slide and tape. Then allow the chambered fly to acclimate at room temperature for 15 to 20 minutes. To generate videos for FLLIT analysis, first turn on the power source and open the viewer application on the connected computer.